You can come in.
Okay.
They're excited. We saw things that look like they belong at Harvard University.
That's exactly right.
Yeah, I do think.
First of all, I know you want to say something to the student, but tell us something about our guest.
Well, this is the 6th annual BCCC Biotech Symposium, and we are so proud. Students do a wonderful job preparing for it, and they're themselves surprised with the outcome they have. And we have so much support from University of Maryland, from the Biopark and everywhere, and people come and support the students, and then they follow up, take in turns. I think this is working out both ways. We have support from administration. Dr. Malaki is always there to support us, and wonderful faculty. So I think this is going fabulous.
Well, one thing for sure, sir, you know this guy right here before.
Right.
What can you tell us about that?
Well, I can tell you that Dr. Watt is an expert in the area of muscle biology, and I think we're going to learn some very exciting things. I haven't worked with him before, so everybody should look forward to his presentation.
Well, one thing for sure, you have an extra smile on your face because I know you're proud of these students who you've seen at the Maryland STEM Festival show off incredibly. What are your thoughts and how do you feel about them now?
Absolutely, and we've been building on this to make it better every year, and you can see the amount of work that's been put in over the last 6 months to produce these works, and I thank the faculty for being the active mentors. Without the faculty, they wouldn't be here doing what they're doing, so I'm very proud of this.
Hey Liz, I can see that you have your shirt already, right?
Yes, sir. Oh, absolutely. I got 2 here.
Get something.
I have my own.
Oh, determination. There it is, a DNA gymnast. I was proud of you of this institution.
This is an awesome institution. I've been here for about a semester now, and it's better than any other institution I've ever been to.
Oh, so you still wet behind the ears with this stuff?
I am.
Sir, you saw some of the posters. What are your thoughts?
Listen, I attended 2 posters already, and after the talk I'll attend many more. I've learned about stem cells. I've learned about algae as biofuels. Really, the diversity of these projects and the quality of the projects is a testament to the faculty and the administration. And as a faculty member and a small business owner and a biotech myself, I'm really excited to explore opportunities with these students as future employees.
Yeah, well, the show is about to start, ladies and gentlemen, so you're not nervous, right?
Not nervous. I'm excited.
Come on, he
I'm
does.
excited.
Every day of it, yes.
So it's almost what time?
Oh, look, make it seem like I'm
We're trying to figure this out together. A gene called BLI1 and a gene called DBY11. BLI1 promotes collagen and DBY11 promotes like body growth and body shape. So what we did was we introduced like a
with like different oxygen concentration, right? So over here, so we use a dog model. So over here, the dog went to cardiac arrest for 10 minutes where they were made dead, right? So they were brought back to life by CPR at different times for 3 minutes.
And on this one, okay. So
This drug was supposed to enhance the drug. So the drug was supposed to enhance the growth of blood.
and we thought that would actually decrease the
So when we add ambulance, we do not want to boost up the oxygen really 100% because it's going to increase the oxygen stress. We do not want to make it just 1% because it's going to
Oh, okay. Okay.
University Community College, it is my privilege to extend a warm welcome to you all. Welcome students and faculty of BCCC. There are professionals from Biotech Company, and we have students from Edmondson Westside High School. We have faculty and students from Hagerstown Community College. We are so excited to have you here and welcome you to the 6th Annual Biotech Symposium, which we call Innovations in Biotechnology. So this event is open to public and is a part of the ongoing Maryland STEM Festival. I hope you guys know about the Maryland STEM Festival. It's like a 13-day event from October 26th to November 10th or 11th. And so we are a part of that event. And so if you are posting pictures, please remember to use the hashtag MDSTEMFestival so that you can show on their Twitter. Thank you. And we are very honored to welcome our keynote speaker for the event, Dr. Chris Ward from University of Maryland. This event owes its support to many people here. First and foremost, the BCCC President, Dr. James H. Johnson Jr., Vice President of Academic Affairs, Dr. Tanya Ringgold, our administrators and supportive leaders. We have our Dean of Division of Natural and Physical Sciences, Dr. Iveha. He could not be here, but Dr. Malaki, who is Associate Dean of Natural and Physical Sciences Department, he's here. Also, we have our SEC President, Dr. Chimalka. We are delighted to welcome you all together, and we want to thank, me and Dr. Kennedy, who has been a relentless supporter of the program as well as the club activities and the symposium. Thank you so much, Dr. Kennedy, for everything, for your encouragement, for guidance, and for your support. And thank you, Dr. Berlin, for being always there for the students. So we would like to share with you that Biotech Club is celebrating 6 years of success. And this year, we are also celebrating 30 plus years of the Biotech Associate degree, so we've been there for a while. Dr. Kennedy has been running the program. And we have the new certificate programs, which are biotechnology and lab animal science certificate programs. As we are in Biopark, I would like to thank Executive Director of Biopark, Ms. Jane Shah, for her support. We are grateful to Dr. John Powers of Baltimore Bioworks for supporting our event with generous donations and things that we needed from time to time. Some of the other people who pitched in to provide that support that we really need to pull this event off. So the lunch of the event was sponsored by Office of Student Life and Engagement of BCCC, so big kudos to them. And lunch is available for you right after the talk, so please do not leave without having lunch. For the success of this event and all the hard work that you saw went into, I would like to ask the BCCC Student Society officers to stand up, and all the members who have put in any kind of work in the symposium. So may I see you, all right? Anyone else who were all at the posters, please stand up. Thank you guys. A big round of applause for you guys for doing wonderful work. And I'm sure some of them are still at the posters. It was hard to kind of, come on guys, we need to go to the top, but I'm sure they're going to be here. So thank you everyone, my family here at home for working with me as we strive for this wonderful, successful event today. And now I would like to invite our enterprising Biotech Club leadership, President Michael Dabney and Vice President Himshila Karti, to introduce the speakers.
Hello, how you all doing today? Give yourselves a round of applause. Hello today, my name is Michael Dabney. I'm the president of the Biotech Club.
Hi everyone, I'm Himshila Karti. I'm the Vice President of Biotech Club.
And we would all like to welcome you all to the 6th Annual Biotech Symposium of 2018. I've been doing this for about 2-3 years now, and it looks good when every time I see all these faces out in the crowd coming to support science. What we would like to do is we're celebrating 6 years of excellence and service to the community of Baltimore. service of the students of the Baltimore City Community College. The prime goal of the club is to help students transition to establish a career in a biotech professional, as a biotech professional, or seamlessly transition into a university for higher education.
So to achieve the goal, the club organizes about 20 plus club events activities in the academic year, including lab tours, talk, inviting scientists, symposiums, entrepreneurs, successful student alumni. The annual symposium, Innovation in Biotech, is the highlight event for this academic year. The club would like to thank everyone who has come here to attend the 6th Biotech Annual Symposium over here.
Now I want to talk a little bit about our keynote speaker. We got a good one for y'all. We're going to uncover a good one right here. So when you hear about this guy, you know you've seen him here first. So let me read it because he has too many achievements for me to remember off top, just too many to name. So I wrote them all down for all his great work that he's done for the field of science. Christopher W. Ward, he's a PhD and he's tenured. He's a tenured professor in the School of Nursing and an adjunct associate professor in the Department of Orthopedics and School of Medicine at the University of Baltimore, Maryland. Dr. Ward graduated with bachelor's and master's degrees from Virginia Tech and a PhD in biomedical sciences, physiology from the Virginia Maryland Regional College of Veterinary Medicine. Dr. Ward did his postdoctoral training with Dr. Mark Snyder in the Department of Biochemistry at the University of Maryland, Baltimore. For the past 20 years, Dr. Ward's research has focused on the mechanisms by which local calcium signals contribute to physiological and pathological adaptations in striated muscle. Research supported by R3, RC2GO, and R1 NIH grants as a PI, several R1 grants as a Code 1, as well as foundation and corporate sponsored research. The discoveries by Dr. Ward and colleagues in heart, skeletal, muscle, and bone have been peer-reviewed and published prestigious scientific journals. The journals that he's been published in are Science, Science Signaling, Nature Medicine, Journal of Clinical Investigation, Nature Communications. I've done a couple biotech presentations from these journals, so I kind of know about these mags. The broad impact of these discoveries is significant, and collectively the results have led to the hypothesis that microtubule alterations are a conserved response to disease stress, leading to altered mechanical transduction and function. Here's the man of the hour.
It is truly my pleasure to be here. I'm going to see if I can get the lights in some orientation where you can better see the slides. So, let me cue this up. So I've got a boring science talk for you. Try to keep it light. This is a fantastic program. I got here early. I attended the poster session. Very impressive. So a big kudos to the students here in the program who are grinding it out, putting in the work. You're going to get rewards. Big hand to y'all. And Edmondson High, you got City in the house. I didn't go to City, but both my boys My one boy's graduated, he's at College Park. My other son is still at City. We took it on the chin in football this year. You guys had us. One last game against Poly. Just see, that's Friday night. All right. So, thanks a lot for having me here. I'll give you the take-home message first, because sometimes when the talk goes long, you don't get the take-home message. So to the students and the students in the program, to the high school students, this take-home message is there are a lot of opportunities in science. Okay? There's a lot of opportunities. You can be a Nobel Prize winner. You could be a research technician in a laboratory. It's all teamwork. Okay? Science is teamwork. So I have a acknowledgement slide. A bunch of people are listed on it. There are too many to count. It's all teamwork. So hopefully someday you'll be on a team and you'll contribute to science, whether it's in academia and industry, what have you. I have a foot in both camps. I'm really an academic, but at the University of Maryland and many institutions, what they really would like is you to discover some things, apply for patents, and then try to spin off an idea to a little company to try to make some things happen. And so I co-founded one of those. It's a very small operation, you know, a couple of us. And so I understand on both ends where you guys can actually fit in, and the future's bright. So keep grinding. So we'll talk about the story, all right? I took a sliver out of a story that we've been working on for, since I guess 2007, and it just keeps improving. The story I'm going to tell you today is work we've done in skeletal muscle and in heart. So you're familiar with your skeletal muscle, right? You're walking around. Motor skeletal muscle of locomotion. You're voluntary controlling that. You're not controlling your heartbeat, right? That's involuntary. You're not controlling your respiratory muscles, your diaphragm. That's kind of just happening. But you can stop your breathing temporarily, right? But then you're going to eventually breathe because you have to, right? So your muscles are really important. And they're a host of muscle diseases. All right, so I've always been intrigued about muscle disease, but really to understand muscle disease, you kind of have to understand how muscle works. So really most of my career has been trying to understand how muscle works. And then if you understand how muscle works, you can maybe understand why it's not working. So I'm going to tell you a little story in that regard. So muscle moves. It contracts and stretches. Your heart is always contracting and stressing. Your vessels are always expanding and contracting. So inside your muscle cells, in every cell of your body, is a cytoskeleton. Right? You've learned about that in biology.
Yes.
Actin, microtubules, intermediate filaments. The muscle has a bunch of other proteins, contractile proteins, that are specialized to make that muscle contract. But all cells have a cytoskeleton. The cytoskeleton does a lot of different things. It transports organelles. It's important for defining cell shape. I just really didn't pay attention to the cytoskeleton at all, because I'm like, it's a cytoskeleton. What could it do, right? It's important for epithelial cells. All those cancer people always talk about the cytoskeleton. Well, now I study the cytoskeleton. And I'll tell you why I started studying the cytoskeleton. So it turns out that there's growing evidence in a whole host of diseases that alterations in the cytoskeleton arise during the disease process, and they act as disease modifiers. That concept is, let's say you've got a disease. The disease is caused by a known entity. That means you have the disease, there's a pressure on a tissue, that tissue has to deal with that disease for a long time, but then the cytoskeleton goes, what's up? I got to change to kind of help deal with what's going on. Those cytoskeletal changes start to make the tissue not behave so well. So they're called disease modifiers. They negatively impact disease and they accelerate disease progression. The diseases that the cytoskeleton has been linked to are cardiomyopathy, heart failure, vasculopathy, lot of vascular disturbances. Neuropathy, neuropathic pain that some patients get with diabetes, foot pain. Chronic pain, but nociceptive hypersensitivity. Critical illness myopathy, respiratory failure in the hospital, and the muscular dystrophies. So the cytoskeletal changes are disease modifiers in these diseases. They might not be the cause of the disease to start, but they contribute to the disease pathology. I have studied for years calcium signaling in muscle. Calcium is critical in muscle. It makes muscle contract. Muscles specialize to handle that calcium. That calcium needs to stay low so the muscle's relaxed, but when the muscle needs to contract, calcium's released, boom, muscle can generate force. But to relax, you got to take that calcium back out. So I've studied the molecular movements of that calcium, and one of your faculty members, Dr. Rotimi Elojo, him and I published a paper together on molecular basis of calcium signaling in muscle. One of your faculty members. So it's also known in muscle that reactive oxygen species. You've heard of reactive oxygen species, even if you don't know you did. Because when you watch TV and you watch those commercials, they want you to drink green tea and they want you to have all these antioxidants, right? You hear all the time, antioxidants, antioxidants. Well, what antioxidants are ways to decrease the reactive oxygen species in tissues, because reactive oxygen species are generated at low levels as signals, but when they're generated too much, they cause disease. So my lab has studied both those things, calcium and reactive oxygen species, and how muscle works. So it turns out that the mechanical stress of the muscle contracting, how those calcium and raw signals are regulated, how the cytoskeleton works, all those converge on one, not so new, but a newer aspect of science, which is called mechanobiology. It's how do mechanical stresses to cells regulate signals. That becomes very important in muscle, right? Because muscle contracts all the time. So not only do we have to worry about where the calcium is going, we have to understand how the forces in the muscle regulate that calcium. So that's what my lab is trying to do for a number of years. We particularly have been interested in those molecular mechanisms. How is that stuff regulated? The title of my talk is about Duchenne muscular dystrophy. So Duchenne muscular dystrophy is a disease. It emanates from the protein dystrophin. So dystrophin is a protein. It's a structural protein. It links the cytoskeleton of the cell to the outside world. In muscle, in this cartoon, in muscle, we've got the cytoskeleton. Here's the contractile apparatus. We've got the actin cytoskeleton. We've got microtubules in the cytoskeleton. They link to the outside world by linking to dystrophin. So dystrophin forms a mechanical link for these cytoskeletal elements to communicate to these proteins in the membrane. These proteins in the membrane link to the outside world and link to the next cell next to it. So when your muscle contracts, forces generated by muscle fibers are transmitted within the fiber, between the fibers. Everything has to be lined up. Everything has to be efficient for things to work. Duchenne muscular dystrophy is caused by a genetic alteration in the gene that encodes the protein dystrophin. And what happens is dystrophin is now absent. And so you don't have that mechanical link. And when you don't have that mechanical link, the cytoskeleton can't efficiently connect to the outside world. And so you have potential problems. So y'all have probably heard of Duchenne muscular dystrophy. If you haven't heard of it, you're probably at the giant food store and the fireman standing there with the boot asking you to stick $1 in. They're raising money for the Muscular Dystrophy Association. It's a very severe disease. The disease arises from a genetic disorder, like I said. There's a genetic problem in the gene that encodes dystrophin. It's an X-linked disease, so it affects boys. So if a boy inherits a defective gene for dystrophin, and can't produce enough dystrophin, or it produces dystrophin that doesn't work so well, these patients develop muscle weakness as early as 3 years of age. So the muscle weakness first is the hips, the upper legs, the pelvis area, the thighs, progresses to the arms, limbs, trunk. By early teens, so in the youngest patients, the babies, you can't really tell. They start to grow, they start to ambulate, they start to walk, and parents often notice they're not walking like my other child, or they're not walking like the rest of the children. And parents start to get concerned, and when they start missing milestones, they'll go to the physician. And there's clinical diagnostics that can determine if this child has Duchenne muscular dystrophy. It's a progressive disease. By the early teens, the patients are most often wheelchair bound, and now, because of palliative treatments and good orthopedic care, the patients aren't dying for respiratory muscle dysfunction so much anymore. What they're dying from is heart failure, because dystrophin is often in the heart too. Well, it's in the heart. Prior, patients were dying because they couldn't breathe. But now the surgical procedures and everything are so good that the patients are living long enough to identify the heart problems. Now they're all dying of heart failure. So it's a devastating disease of the skeletal muscle and the heart. And it's a progressive pathology. The patients are born without dystrophin. The children look normal up until the time they ambulate, and it gets worse and worse and worse and worse over time. So, therapeutic options. The gene for dystrophin was discovered in 1987. They knew the gene in 87. And you go, why not do a genetic therapy? Just replace the gene. That'd be fantastic. It's a massive gene. When you start to look at genes and you start to understand these genetic therapies, this gene has about 2.4 million base pairs and 79 exons. It's a massive gene. It's not that big of a protein, but it's a really big and complicated gene. So the size is a major challenge. You just can't put that in a virus and try to deliver it. That's a problem. There are therapeutic options coming down the pathway. There is opportunities to skip damaged exons. You can use genetic therapies, AAV therapies, to try to skip those exons. Those are in clinical trials. You can deliver smaller genes, truncated proteins, or surrogate proteins to help that structure. Some of those are in clinical trials. But the horizon is far away for the majority of these patients. They are declining in function daily. So, I'm not a gene jockey. I'm not a molecular biologist. I don't do gene therapy. What I'm interested in is disease modifiers. Those patients didn't have dystrophin from the day they were born, and the disease gets progressively worse. So the likely are changes in the cell that cause that disease progression. If we could find them and target them, maybe we can slow disease progression so that the kids are around when genetic therapies are developed. That's the goal. So identifying disease modifiers to halt or slow disease progression and also enhance the efficacy of genetic therapies. This is a goal for a lot of genetic diseases nowadays. When you get into science, you'll realize either you want to cure it or you want to dampen down its impact. So when you're thinking about attacking a problem, you can go one or two ways. My lab has gone the latter, identifying the ongoing problems to try to slow disease progression. Here's what we know. We know in muscle, stretch and contraction elicits these mechanical signals. It elicits mechanical activated ROS signals from ADP oxidase 2. It's an enzyme that produces ROS as a signal. Those mechanical signals also make calcium channels open, and they make calcium signals. That regulates normal physiology in the body. It regulates transcription, mitochondrial function, glucose uptake, autophagy, lots of things in all cells. And muscle regulates contraction. But if you have a dysregulated excess in those signals, if those signals are way too exuberant, that drives pathology. Excess calcium and excess ROS have been linked to a suppression of these key cellular functions, which drives disease. So what we have been interested in is the mechanisms responsible for this dysfunction. How are the calcium channels activated by mechanical stress? How is the NADPH oxidase 2 activated by mechanical stress? If we knew that, then maybe we'd understand the disease better. A whole host of diseases better. So we got to that work to try to understand that. So rather than plow through a bunch of slides, I'm going to give you the end. This is the end. We discovered a mechanotransduction pathway. So I used to always think at simple little levels. I would think, oh, I want to know what calcium does. I want to know what this does. I want to know what that does. But then when you want to understand how mechanical stress alters these signals, you have to explain how mechanical stresses are interpreted by calcium channels or enzymes to do something. So mechanotransduction pathways are pathways by which mechanical signals are transmitted to channels and enzymes to work. So what we have discovered through a number of years is that stretch or contraction in the heart or skeletal muscle, or more recently, in just mechanical loading of bone, we've shown that mechanical signals transmitted through the microtubule network, a critical component of the cytoskeleton. Those mechanical signals through the microtubule network somehow, we don't know how yet, but somehow activate this enzyme, NADPH oxidase 2. This enzyme produces ROS. Since it's ROS produced by NOX2, we've called it X-ROS, so that it's distinct from all the other ROS sources. It's mechanoactivated ROS. That ROS, we show, targets calcium channels. So we have a pathway. And when you have a pathway and you understand this in normal cells, you can go, all right, well, let's see how this plays a role, this plays a role, this plays a role. You can start mucking around with things and figuring it out. So this is the culmination of a whole bunch of work. Started in 2007-ish, published our first paper in 11. Our last paper was 2017 in Bone with my colleague Joe Staines in the back. So we've been working on this a long time across multiple tissues. In muscle, here's how we think this whole thing works. So this is a striated muscle cell. This is the outside of the cell. This is the inside of the cell. In the inside of the muscle, we've got a mitochondria. We've got some contractile proteins. We've got an organelle that stores calcium. And we've got some channels. We've got a calcium channel here and a calcium channel here. And we've got our NOX enzyme. And we've got these little cartoon microtubules. Microtubules connect to dystrophin. Here's our membrane complex, and we've got the extracellular matrix on the outside. So our idea is, in cartoon form, with stretch or contraction, that activates force through this complex, activating force through dystrophin, tugs on the microtubules. The microtubules somehow tug on NOX2. That makes them produce ROS. That ROS gets into the cell, targets by oxidation calcium channels, TRP channels on the outside, rhinoreceptors on the inside to regulate calcium release. Now, that is an effect in healthy muscle, heart and skeletal muscle, that helps the muscle work over time. So we wrapped up that story. We're pretty happy with it in the muscle. It's a fascinating twist in bone. I don't have time to go after that today. We're going to stick with the muscle. So how about just muscular dystrophy? How are we going to swing back to that? Well, we've known long ago that a dysregulated excess of mechanoactivated NOX2 ROS and calcium influx were early events in DMD pathology. And that caused enhancement in muscle damage that drove the disease process. But not too long ago, a paper from now a colleague, Jim Ravasti, showed dystrophin itself is a microtubule binding protein. We didn't know that before. And when dystrophin is gone in Duchenne muscular dystrophy muscle, that causes the microtubule network to be highly disorganized and to get much more dense over time. So we get a densification with disease progression. So we've got the calcium problem, we've got the NOx2 problem, and now we've got a cytoskeletal problem that appears as if it tracks with disease progression. So you go, well, maybe microtubules are part of the mechanotransduction pathway and driving this dysfunctional signal as the disease progresses. Maybe. So let's test it. So in our minds, Duchenne muscular dystrophy was an empirical test for what we discovered in the skeletal muscle and the heart. And if we learned something, then maybe we had a disease-modifying strategy. So our model system that we make most of our discoveries are single cells. So we take either a heart out of a mouse or a skeletal muscle out of a mouse, and we put them in enzyme, we isolate their single cells. So this is a single cardiomyocyte, and this is a single skeletal muscle fiber. We can keep them in culture conditions for a day or two, keep them happy, and then we get studied. So we can take these from healthy animals, animals with Duchenne muscular dystrophy, all kind of different animals to study physiology. So what we do with these is we attach them to a force transducer and a length controller, and that allows us to grab these little cells and to stretch them, or to ask them to contract and measure their force. So we can study all this stuff at the single cell level. This equipment is over a microscope, so we can load these cells with dyes that track calcium or reactive oxygen species, right? So we can measure those signals when we stretch. So if you look at the business end of this apparatus, there are little glass elements, and those glass elements, when you zoom in, look like this. So we worked with a company to develop these. So these have a little channel on the bottom, and we coat them with a glue that we developed at the University of Maryland. And we can go down and we can stick our skeletal muscle, we can lift it off the dish, and we can stretch it, we can ask it to contract, we can do a bunch of things. So, microtubules. Microtubules are dynamic structures. These are cytoskeletal elements made from tubulin protein, and that tubulin protein occurs in dimers. One protein interacts with another protein, make a dimer, and those dimers polymerize. We're in the depolymerization phase here of this movie. So it polymerizes to make the filaments long, and on the other end, it's depolymerizing to make it short. So these structures are always dynamic in the cell. All right. So here's a dynamic structure. It's growing on one end. Here's free tubule. It's growing. The tubulin's being added to this. It is an intermediate state, and then it's also coming apart. Now, we've long known, because of cancer chemotherapeutics, that you can manipulate these structures. There's a drug called Taxol, or paclitaxel, a cancer chemotherapeutic, first-line chemotherapeutic for a number of cancers. That drug causes microtubule polymerization. It causes the microtubules to polymerize, and the microtubule network gets very dense in cells. There's also a drug called colchicine, or Colchris. This is a frontline drug for gout, gouty arthritis. It's also used in everyday therapy for patients with familial Mediterranean fever. And it's been shown important in some heart diseases. All thought to be due to its anti-inflammatory effects. But we've got drugs that we can make the microtubules more dense, and we can make them less dense. We can depolymerize them. These are FDA-approved drugs. So here's our little rig. We attach a muscle fiber, we apply a length change to it. We can measure that length change by measuring the muscle itself. And if we load that cell with a Ross indicator called DCF, what you notice is when we start stretching that cell repetitively, here's the level of Ross, it's quiet, 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 and we start stretching, boom, it starts producing Ross. And we stop stretching it, boom, the Ross shuts off. So we've got mechanoactivated Ross. We can measure it, we can figure it out. We can measure calcium influx. So as soon as if we load a calcium indicator dye and we have a cell at rest and we start stretching the cell, calcium starts to go into the muscle. And then it flattens out when we stop. So we can measure those two very important signals that are dysregulated in Duchenne muscular dystrophy. Excess calcium influx and too much ROS. But these are healthy cells. Okay? And here's a diagram of what those cell signals look like, and here's a diagram of a skeletal muscle fiber with the microtubule network labeled with an antibody. So we're labeled alpha tubulin, we can see the network. There's a single skeletal muscle fiber. In Duchenne muscular dystrophy, the animal model, which is a dystrophin-deficient mouse, it looks like this. So the microtubule network is much more dense. So if we have a much more dense microtubule network, and those microtubules are important in generating these mechano-signals, what do you think might happen? The signals might be more robust, right? They might be more exuberant. Sure enough, we've quantified it. So in these cells, the microtubule density is elevated. The mechanoactivated ROS is elevated. The mechanoactivated calcium influx, way elevated. So we've got a gain in these signals in the MDX mouse. But we already knew that, right? We kind of already knew that. We didn't know how microtubules were involved though. So let's take that drug colchicine off the shelf. Let's treat these fibers with colchicine. Here's this MDX fiber that you treat with colchicine for an hour. We've taken down the density of those microtubules, right? And now let's stretch them. We take the density down, we wipe out the stretch-activated ROS, and we wipe out the stretch-activated calcium. That's gained. So, proof of concept, the microtubules seem to be involved in these excess mechanoactivated signals in the muscle. In the muscle fiber from the foot of an MDX mouse. Fantastic. So we think microtubules are potentially a disease modifier. Now, you go, well, do you really know it's a disease modifier? If it's a disease modifier, you should be able to take Taxol and take a healthy fiber, put Taxol on it. Let the microtubule network get really, really dense, and then go, if microtubules really were involved, now those signals should look like the disease. Sure enough, the calcium influx is elevated, the stretch-activated ROS looks like MDX, and that density as high as the MDX. So this is a conglomeration of results from across two papers, that established the fundamental basis for this finding, that microtubules appeared to be critical for these excess mechanoactivated signals in the muscle in muscular dystrophy. So we know as microtubules are dynamic structures, sure the density is important, but these proteins are heavily modified by post-translational modifications. That being that protein is made, and then it's modified somehow after it is made. Specifically, microtubules are heavily post-translation modified by a number of things. We were particularly interested by a post-translational modification called detyrosination. Detyrosination is when there's an N-terminal tyrosine on tubulin that gets cleaved. Why were we interested in that? Well, this is a skeletal muscle fiber. Instead of just stained with, it's a wild-type fiber. Instead of being stained just with for alpha-tubulin, like you saw on the slide before, we stain for alpha-tubulin green and detyrosinated tubulin in red, and we see that PTM abundant in the wild type. And if we do some fancy microscopy, super resolution microscopy, and zoom way in, we can really see these structures. But why were we excited? Well, this post-translation modification makes microtubules link to other structures, makes that cytoskeleton really transduce force in the cell. So it's likely that this post-translational modification was critical for the mechanosignaling with or without that increase in density. And what we had known after collecting a bunch of data, that the abundance of this PTM by Western blot, so here's the alpha tubulin wild type, here's the alpha tubulin MDX, we have a really huge increase in the abundance of this post-translational modification to tubulin in a number of dystrophies. Not only Duchenne muscular dystrophy, but dysferlinopathy and desmonopathy. So we thought, this might be an interesting target. Well, it's an interesting target for a number of reasons. Number one, there are drugs that you can manipulate it, right? So the carboxypeptidase is an enzyme that takes this terminal tyrosine off the tubulin tail and clips it off. That yields a terminal glutamate that was termed glutubulin, but there's also glutamylated tubulin, and that's kind of confusing. So now some individuals start calling this D-tier tubulin or detyrosynated tubulin. So detyrosynated tubulin has this terminal tyrosine gone, but there's an enzyme called TTL, tubulin tyrosine ligase, that can stick this enzyme back on. So it's a cycle that happens in the cell for reasons. We reasoned that could we specifically target that detyrosynated tubulin and leave the regular tubulin intact? So here's control cells, alpha tubulin, stained for detyrosynated tubulin. Here's the overlay. If we apply Taxol, we get an increased density of the microtubules, extra bundled microtubules with detyrosynation. If we put on that PTL drug, the microtubule density doesn't appear to change, but the amount of detyrosynation is wiped out. So it appears as if we can reduce that post-translation modification, but not mess with the structure of the microtubule network at all. Okay? And then we quantified it to show it was true, but I'll just move on. You can read the paper. What we knew also, because of our work with Stuart Martin in the Cancer Center, is that Taxol increased this microtubule density and detyrosynation. So part of our result before could have been was detyrosynation as well. And that PTL treatment decreases the density of detyrosynation without altering the microtubule density. So then we did the experiment, and these are in wild-type muscle cells. These are healthy muscle cells. The question was, if we did our same experiment, if we attached a muscle fiber, applied stretch, measured ROS, and we did it in control conditions, if we treated these animals with PTL, the muscle fibers with PTL, to decrease detyrosynation, but the microtubule density did not change at all, we wiped out the mechanostigma. Just like when we completely wiped out the microtubules. But we didn't wipe out the microtubules. Just that post-translation modification. So indeed, that post-translation modification caused microtubules to link to other elements to transduce force. You take it away, the microtubules are there, but they can't do their mechanotransduction job. So we thought that was fantastic. We did it pharmacologically with PTL, and we also did it genetically with TTL. So if we overexpress TTL, we also drive the decrease in that post-translation modification. So we did it genetic pharmacologically. And we used a peptide inhibitor of the NOX2 ROS enzyme to show that that was equally effective. So it's within our pathway. So I'm going to skip the data. So the effect of PTL is independent of density. So now you're going, all right, what's the pitch, man? This guy's at the cell, he's doing all this crazy stuff. What's the pitch? Is this even possible for any real life importance? So you got it back to the whole mouse, right? So when you go back to the whole mouse, you say, what is the established clinical endpoint in muscular dystrophy in these preclinical models such as mice? Well, it's contraction injury. It's the susceptibility to contraction injury. And the contraction injury that we do is called eccentric injury. So we have a mouse here. It's anesthetized with isoflurane gas, just like they have in the OR. So he's laying there, anesthetized, doesn't know what's going on. We didn't do something barbaric to his leg. This picture is, we used hair remover, Nair hair remover. Yes, I went to the drugstore and bought like a huge 3 bottles of hair remover and a bunch of gauze, and the checkout person's looking at me like, sell. So we bolted the leg of the mouse so we can see where we can put our electrodes. And then what you do is you do a eccentric contraction. So you deliver a contraction, so you make that foot contract. And that foot's in a foot plate, and that's connected to a force transducer. So we can measure how much force that foot is producing. But the animal's completely intact. The blood flow's intact, we're stimulating the nerve. But we're doing it. The mouse isn't doing it. We're stimulating the nerve.
And so if we give a full contraction, once the muscle's contracted fully, we stretch it. And you go, how's that possible? Well, you do it all the time. When you're up on the 6th floor and you're coming down the stairs, right? I can't do it, but I'm terrible at it, but if you're coming down the stairs, right? When you walk down the stair, you're doing a braking contraction. Your muscle's contracting, but it's lengthening at the same time. So if you're in the gym, right, and you put on more weight than you can really push up, and you're contracting while it's coming down, right? A negative, that's an eccentric contraction. Those contractions are the things that cause soreness, right? So you go for your weekend hike, it's not the hike uphill that gets you, it's the hike back downhill. And that night you go, ooh, I think I'm gonna be sore, right? You go, oh, I think I'm gonna be sore. And you're a little sore. Then you wake up the next day and go, yikes, I'm really sore, right? 2 days after, you're like coming to work late, right? That's called delayed onset muscle soreness. That's because you had some muscle damage. So we can do that in the mouse. Here's our first contraction. Here's our 20th eccentric contraction. So we lost force. We've slightly damaged that muscle. And it takes days for it to recover, a couple of days. If you do the same thing to an MDX mouse, those same 20 contractions, there's the first one, there's the last one. It's really susceptible to this injury. Partially because of the calcium, the reactive oxygen species, dystrophin's not there, it's easily damageable. So you can use this as an in vivo assay. Right? So what do? Here's our little pathway that we talked about. Force through microtubules, stabilized by detyrosination, alters stiffness, drives up mechanotransduction to ROS and calcium. In disease, all these things are out. So if we take an MDX model of Duchenne muscular dystrophy, we can treat that mouse with colchicine, because it's a drug that people take for gout. We can also treat that animal with LC1, which is a prodrug of parthenolide. Now LC1 is not FDA approved, but it's in clinical trials in Europe for GI cancer and brain cancer. But we can treat the animal with it. We can treat these animals, and then we can test them. So, here is a wild type animal. Here's the first contraction. We've normalized it to 100, and here's how much it drops over 20 contractions. Here's the MDX. Okay, 20 contraction, it drops to like 20% of its peak force. The animals pre-treated with colchicine for 4 hours were significantly protected from this force drop. The animals treated with LC1 for 3 days because of the pharmacology of it, significantly protected just like colchicine. So we conclude that, yes, indeed, targeting microtubules with either FDA-approved drugs or new drugs that are coming on the market might be beneficial to delay the muscle damage in Duchenne muscular dystrophy. That was ours. So, you say, what about the heart? The kids are dying from heart failure, right? We can do that. We've done those experiments in the single cell level. I don't have time to show it. But here's the in vivo stuff, which is kind of cool. So to do heart, what you do is you anesthetize that animal. I can't have them run on a treadmill and monitor stuff, right, and behave, so I just anesthetize them. And I give them a drug to increase their heart rate. They do this in the hospital all the time. They don't use isopaterenol, they use dobutamine, but it's called a pharmacologic stress test. The person's laying there, the mouse is laying there, you inject them with the drug and heart rate, whoop, goes up, stays up for like an hour, and then slowly comes down. In the animal, here's the MDX treated. This is an experiment with parthenolide. So MDX treated baseline, 3 minutes post isopaterenol injection, the heart rate goes way up. This is the EKG, so each of these is a heartbeat. The resting heart rate of a mouse is about 400 beats per minute. Under anesthesia, it's about 200. So this thing went up to 800 or 900 with isopaterenol. After about 35 minutes, it started to really drop because we had some QRS complexes, but they went away. So we got complete failure of that heart. And some of these animals started to die. The MDX treated with parthenolide, still cooking. Doesn't seem to care. 40 minutes post, Here's an example of arrhythmogenic death, and we took the animal off. Parthenolide, animal keep cooking, no problem. So the best way to explain this is through a live dead thing. So here's MDX animals, 9 of them treated, 9 of them untreated, 0 survived the protocol, 9 of them died. If we pre-treated these animals with parthenolide, much like we did with the skeletal muscle, 12 of them lived, none of them died. If we muck around with other parts of our pathway, right? If we just target the NOX2 by treating these animals with GP91 DS-TAT to decrease the mechanoactivated ROS, remember that ROS targets the calcium channels. 5 of them survived, 1 of them died. Not bad, not bad odds. Treat them with colchicine, 11 of them survived, 1 of them died. Treat them with a different type of calcium channel blocker, our most recent publication. 8 of them survived, 0 of them died. So if you target this pathway, it prevents against arrhythmogenic death or workload-induced death in the dystrophic heart. And it highlights the pathway. You can target the microtubules up top or any of the components on the bottom and get the benefit. But we're fancy on the microtubules. We like them the best. Is this a clinically translatable target to humans? Difficult question, right? You can't take a heart biopsy from a DMD patient. Skeletal biopsies are hard to come by because there's genetic tests and they don't do biopsies in the boys much anymore, but you can get these samples. We had a grant to get some stored samples from a group, and we did, in collaboration with the group upstairs and IGS, some transcriptomics. So RNA-seq analysis of the transcriptome between human DMD boys and non-diseased boys, their clinical muscle biopsy samples. And we did transcript. So here, red is elevated transcript, blue is decreased. It's a mess to read, but here's the upshot. If you look at microtubules, we've got an increase in transcription of the microtubule axis in the DMD. We've got an increase in transcription of the NOX2 axis in the DMD. Increased transcription of the calcium signaling axis. What's of interest to us now in work across our tissues is that comes with a decrease in the ability to buffer that toxic ROS. So we think this pathway is important in the human. Here's the conclusion. We think microtubules are likely a mechanotransduction signal that regulate NOX2 ROS and calcium. Microtubule mechanotransduction is excess in dystrophic skeletal muscle and heart. We propose that the microtubule network is a novel targetable drug space for disease progression of muscular dystrophies. That's our story in the muscular dystrophies. Thank you. And I'd be happy to take any questions now, or if you're shy. If you're shy, ask them. If you're really shy, you can. No, this is diagnosed usually at milestone failure. Question?
Great talk. Since there is a difference between animal muscle and human muscle, have you tried growing animal muscle in the mouse and testing it that way?
Fantastic question. There is a group on campus here, and there's groups on many campuses that are taking human myoblasts or human iPSCs from these patients, from a lot of different diseases, growing them and planting them in mice to try to get humanized muscle in the mouse, and that works. Right, you can do it. For a disease like this, it's the effort is not really worth the gain because the protein functionally works the same in both. Now there might be some gain because the signaling might be different, and the humans are more susceptible. These mice, they have a relative resistance. The upshot is your standard FDX mouse lives a virtually normal life. And you're like, well, why do you study it then? Well, it's important because we can
and study its trajectory early on, the MDX mouse has been improved by crossing it to make it more humanized, and that animal has a short lifespan. So we use multiple animal models, but for this disease we haven't gone to like xenograft work.
What's next for your group?
Next for our group, we are doing, we have parallel projects in, currently funded projects in this, right? We believe the same pathway contributes to the development of atrial fibrillation. So you say atrial fibrillation nowadays, people are like, what? Everyone, it's like every time you turn around, it's like everybody's in AFib for reasons, right? And there's lack of drugs to be able to do that, but AFib is a mechanotransduction problem, and so we're looking at that with the grant. And then we have some of our newest work, and it's fantastic. And I'm kind of like a technical person and go, yeah, I love it. But who's driving the project is my colleague in the back, Joe Staines, in bone. So it turns out that when this pathway is dysregulated, in muscle, the signals are too much. In bone, when microtubules are dysregulated, there's too little signal. For a reason you read about in paper. But when there's too little signal, the bone appears as if it thinks, I got plenty of bone. So then it underscores osteoporosis. The cell says, I have plenty of bone around me because it has more cytoskeleton than it should. But then the bone crumbles around it. So we're trying to figure that out. So that's what we're doing across a couple of questions.
Yes. On your RNA sequence slide, the inflammation, I noticed you had inflammation cycle. What role do you think that is playing in Duchenne dystrophy, and what role do you think it may play in atrial fibrillation?
Yeah, that's a great question. That's a good eye too. I hit it quick to go by it. I actually didn't hit it quick to go by it. The answer to your question is a partial answer to your question. The big open question is, why do you get these microtubule problems? It turns out when the Duchenne, when the MDX animal is really, really young, it doesn't have a microtubule problem. The microtubule develops with disease. So your next question is, what is driving the microtubule problem? So it turns out that it looks like what's driving the microtubule problem is reactive oxygen species and inflammation. And so inflammation also drives reactive oxygen species. So it's like the hyperinflammatory environment seems to be pushing cells toward this microtubule disarray. And so we're trying to figure that out in a couple of our tissues.
And if you took multiple passes of that same cell,
Yeah.
would you get generational or age answers?
So the answer to that question is, I keep saying our latest work, our newest work, our yet unfunded work that we're putting in funding for, it seems as if these changes track with the development of sarcopenia, which is age-related muscle decline.
Right.
And so we think there is an age effect, right? There's a disease progression and age progression, and everything is linked. So yes, we think there's an age-dependent effect, and we're working on that too.
So maybe this is what John was getting at. So when you were talking to your therapy and the recovery, so normal cells will recover after like 48 hours or whatever, and the MDX, do they, because they're so much more knocked down, do they recover at all, or do they just stay knocked
The MDX
down?
mouse will recover, but like if we do this really mild protocol, 20 contractions, the wild type will recover in like 3 or 4 days. The MDX will take about 10-ish to 14 days if they recover. But the MDX mouse is not like a boy. The more humanized mice that really get injured, you injure them like that, they'll take them 3 weeks to come back to half. It really drives them down.
So is age progression sped up in muscular dystrophies, or?
That's a great question too. That's submitted a grant to NIH, and I kind of use these terms about accelerated aging and disease, and they just hammered me. But fundamentally, if you think of aging not as chronological aging, but biological aging, our work shows that a 2-month-old MDX mouse muscle, its microtubules, look like the microtubules of a mouse that's about, a healthy mouse that's about 14 months old.
Any links
So you say
between-
that could be like a accelerated aging, sure.
Yeah. Any links between like progeria, I don't know if I'm pronouncing it right, and muscular dystrophies or anything?
I'm going to turf that question because I know nothing about progeria. I'm fascinated by it. Some molecular aspects of progeria, signaling aspects. My colleague who studies Bode, Joe Staines in the back, he has animal models of progeria deficiency. So there is a fascinating little link about some aspects, but it's beyond my pay grade. Question?
Do you imply that dystrophy is somehow related or plays some important role in inflammation?
That's an interesting question.
Because you just mentioned
Yes. So, we have no evidence that dystrophin plays a role in inflammation, but if I really simplify it down for you and I say, if dystrophin's absent even in a young animal, and that animal is more susceptible to contraction injury and muscle cells damage, and they release some of their constituents, that seeds an inflammatory event, and then over time, more cell damage, then more inflammation. So the frontline treatment for boys with Duchenne muscular dystrophy has been steroid anti-inflammatory therapy. The problem with that is that degrades muscle, tendons, organs. So as soon as you go high steroids, you.
I know we have more questions. I would want you to meet some exhaustive us for more questions, but that was a
Here all
wonderful talk.
afternoon.
Well, I want you to just stay here for like just 1 more minute. I want you to slide the board.
Just a huge small gift of thanks to you, to Baltimore State Community College.
So we're going to take one, go to the other end of the table, get you some chips and soda.
First of all, Dr. Kennedy, here at the Biopark, you certainly are proud of what you saw with these students.
I'm very proud of our students. They make us look so good for the Biotech program and the Life Sciences Institute and the Baltimore City Community College.
I can tell you one thing, sir. I can pose this question to you. What are your thoughts of what you saw in there with these students?
Well, I'm an environmental engineer, so I was really amazed at the topics they're looking at and insights that we're gaining through their research. So I think that we're starting something big here. Getting students who are at the community college involved in research will take them to the next levels because of their hunger to want to be able to discover things and make things better.
I can tell you, sir, they enjoyed that conversation in there. It was enlightening, intriguing. What are your thoughts?
I really enjoyed giving the talk. The treat for me was the questions afterwards.
Yeah.
The students asked really insightful questions. Really insightful questions. And learning afterwards that really in the audience not only are high school students, students from this program, so future students who are coming in your program, and members of this Biopark community who are leaders in the field, leaders in the corporate field. So I know they're thrilled to have these students as team members.
Sir, you saw your staff, Dr. Kennedy, Dr. Berlin, Dr. Benagucci. How proud are you of this team? Being doctors and all, they have a special thing for biology. How special?
This is really special, and we've been working for the last 5 years, and you can see every year we're doing better than the previous year. And thanks to the two of our great faculty we've had here.
So, sir, you can give us a prediction on next year. Is it going to be harder? Because this is a tough thing to beat out.
I think what you're going to find next year is more maturation in the students' research, taking it to the next level. I think what you also will see greater creativity because now they're being encouraged to think about not necessarily what everybody else has thought about, but how about some new approaches to do the research that they're doing. So bigger and better is what I would say.
Good.
I have to say that students work so hard for this, and this is all for students. So I'm thrilled you gave a wonderful talk and for your encouragement that you came all the way for this event. So thank you so much.
And before you leave, Dr. Johnson, do you...
Yeah, that was one of the interesting projects. Like,
No, but I mean, I read this thing, I know what this is. Right? Plastic party cups. Why did they could have just said, plastic drinking cups or whatever.
I will have to give it like scan or something.
Sure, okay, fine.
How you doing, sir? You enjoying this event?
Yeah, pretty good, thank you.
So Mr. Lewis is our adjunct faculty in the Biotech program. He's a wonderful job teaching 2 online classes. Yeah, and you've been associated with the program before, right?
Yeah, I was a volunteer teaching assistant.
But besides that, you also work. So
Yeah, I'm also I have a full-time job working at a small medical devices company here in Baltimore. So I just took some time off to come in here quickly, take a look at these awesome posters, see the cool stuff. Thank you.
So, I mean, it's easy to remember.
thank you so much, sir.
Okay, yeah.
So this work was done where? Where did you do it?
So this summer I worked on the University of Maryland lab, and I was looking at the impact of different levels of oxygen on the cardiac arrest on the brain, right? After cardiac arrest on the brain. So when the cardiac arrest happens, there's like oxidative stress when we give them oxygen back to them. So in that thing, when there's oxidative stress, there's NRF2 and hemoxygenase are the protein. They translocate to the nucleus of the cell. Then they go to some reactions and they produce hemoxygenase, which is like an antioxidant. So they counteract against antioxidative stress. So over here we are trying to see like what concentration of oxygen is good on the brain after having a cardiac arrest.
Okay.
So
what was your findings here?
So in my thing we use a female beagle dog. We underwent the cardiac arrest for 10
Why did you
minutes.
prefer the dog? What was the reason for the
Because
dog?
we already did in mouse, so now we're going a little bit higher.
Higher level.
Okay, so 10 minutes, they went to cardiac arrest for 10 minutes. Then they were brought back to the life with open chest CPR for 3 minutes with 100% oxygen. So, because they didn't have oxygen before, so we provided with 100% oxygen. So cardiac arrest doesn't usually happen in hospital. It happens in exactly. So transient phase, we name that as the ambulance phase, and this one is the hospital phase. So in the transient phase, like we are kind of confused, like whether to give them normoxic, whether to give hyperoxic, but so.
Very good.